Indian red scorpion, and the venom behind sudden collapse. A farmer in the Konkan region of western India finishes a long day of plowing fields beneath a punishing sun. Night falls. He kicks off his sandals and lowers himself onto a thin mattress on the floor of his one-room home. His children are already asleep beside him. The air is warm and heavy. Crickets hum through the darkness, and somewhere in the crumbling wall, just 3 feet from his pillow, a creature no longer than a grown man's finger is stirring, shaking off the stillness of the day, flexing pincers the color of burnt orange. It moves silently across the earthen floor. It does not seek the farmer out. It does not hold a grudge. It is simply following the scent of the cockroach that slipped beneath the mattress earlier that evening. Then the farmer rolls over in his sleep. His bare foot presses against something hard and smooth. In less than a second, a tail arcs upward and drives a needle-sharp stinger into the arch of his foot. The pain is instantaneous, a white-hot jolt that tears him out of sleep and into a waking nightmare. Within 15 minutes, he is drenched in sweat. Within 30, his heart is hammering so wildly he can feel it thudding in his throat. Within the hour, his lungs begin filling with fluid, and each breath becomes a wet, desperate gurgle. If he does not reach a hospital with the right medication, he will be dead before sunrise. That is the reality of living alongside Hottentotta tamulus, the Indian red scorpion, an animal that some researchers have called the most dangerous scorpion on the planet. Not because of its size, not because of its aggression, but because of what is hiding inside that tiny, amber-colored tail. A venom so precisely engineered by 400 million years of evolution that it can hijack the entire human nervous system in minutes and send the body into total cardiovascular collapse. Here is a fact that might keep you up tonight. Out of more than 2,500 known species of scorpion crawling, burrowing, and hiding across 6 of the 7 continents, only about 25 produce venom strong enough to kill an adult human being. And of those 25, the Indian red scorpion consistently ranks at or near the very top of every lethality list. Before the discovery of a proper treatment protocol in the 1980s, the fatality rate from its sting ranged between 8 and 40% in clinical studies. In certain rural pockets of India, it was even higher. That means if you were stung in a village without access to the right drugs, you had roughly a 1 in 3 chance of dying. And the most heartbreaking part, the majority of those victims were children. But what makes this particular scorpion's venom so devastatingly effective? Why does a creature that weighs less than a handful of coins possess a chemical cocktail capable of dropping a full-grown human into pulmonary edema? And how did a single doctor working out of a tiny primary health center in rural Maharashtra manage to slash that terrifying death toll from 40% to less than 1%? armed with nothing more than a blood pressure pill and stubborn, relentless courage. Tonight, we are going deep into the world of the Indian red scorpion. We are going to trace its ancestry back to the primordial oceans of the Silurian period, more than 430 million years ago, when its earliest ancestors crawled through shallow seas alongside trilobites and jawless fish. We are going to explore the astonishing biological machinery that makes its venom one of the most targeted chemical weapons in all of nature. We are going to walk through the rural landscapes of India, Pakistan, Nepal, and Sri Lanka, where this scorpion lives in terrifyingly close proximity to human families. And we are going to meet the people, both scientists and village doctors, who have turned a story of death into a story of hope. So settle in, pull the covers a little closer, and whatever you do, make sure you know exactly what is on the floor beside your bed. To understand the Indian red scorpion, you first need to understand just how ancient the scorpion lineage truly is, and ancient does not begin to cover it. When we say scorpions are old, we mean they are staggeringly, almost incomprehensibly old. They predate the dinosaurs by more than 200 million years. They predate the first trees. They were skittering across the earth before anything with a backbone had figured out how to breathe air. The oldest known scorpion fossil in the scientific record is a species called Perioscorpio venator, unearthed from the Brandon Bridge Formation in Wisconsin. Of all places, the fossils date to the early Silurian period, roughly 437 million years ago. When those creatures were alive, Wisconsin was not the cheese-making heartland we know today. It was a warm, shallow ocean floor at the base of a small island cliff, blanketed in silt and populated by worms, early arthropods, and the very first hints of life preparing to leave the water. Here is what makes Perioscorpio so extraordinary. When paleontologists Andrew Wendruff and Lauren Babcock at Ohio State University examined the fossils under high-resolution microscopy, they found something that, frankly, stunned the scientific community. Preserved within the rock, pressed into the thin dorsal cuticle by hundreds of millions of years of sediment compaction, were the outlines of internal organs, a gut tract, structures resembling a vascular system, and most remarkably, hourglass-shaped chambers that appeared to house the scorpion's respiratory system. And those internal structures, they were essentially identical to the respiratory and circulatory systems found in scorpions alive today. Think about that for a moment. The internal plumbing of a scorpion living in a shallow sea nearly 500 million years ago is, in terms of basic shape and layout, the same as a scorpion you might find under a rock in Mumbai this evening. Babcock himself put it plainly. The inner workings of the respiratory circulatory system in Perioscorpio are shape-wise identical to those of modern scorpions that breathe air exclusively, but also incredibly similar to what we see in marine arthropods, like horseshoe crabs. In other words, scorpions appear to have been pre-adapted for terrestrial life. Their bodies were ready to breathe air before they ever left the ocean. This discovery answered one of paleontology's most persistent questions. How did scorpions make the leap from sea to land? The answer, it turns out, is that they barely had to change at all. Their respiratory system, a series of lungs tucked inside the body that function much like the gills of a horseshoe crab, worked in both environments, while other lineages had to undergo dramatic evolutionary overhauls to survive the transition from water to land. Scorpions simply walked ashore and kept doing what they had always been doing. It is one of the most elegant examples of evolutionary pre-adaptation in the entire fossil record. And it explains, in part, why scorpions have survived every mass extinction event in the history of life on Earth: the Endordovian extinction, the late Devonian extinction, the catastrophic Permian-Triassic extinction, which wiped out more than 90% of all marine species and roughly 70% of terrestrial vertebrate species, the end Cretaceous extinction that erased the non-avian dinosaurs 66 million years ago. Through all of it, scorpions endured. Their body plan was so efficient, so fundamentally well-engineered for survival, that it required almost no modification across geological ages that saw entire ecosystems rise and fall. Now, Perioscorpio was not quite the scorpion we would recognize today. It had compound eyes, similar to those found on insects and crustaceans, positioned at the front of its head. Modern scorpions have traded those for simpler eyes, a pair of median eyes on top of the carapace, and additional lateral eyes along the sides. Perioscorpio also had 7 sternites, the belly plates on the underside of its body, whereas modern scorpions have 5. Over hundreds of millions of years, there was a progressive reduction in that number. But the clawed pincers were there. The segmented tail was there. And most critically, the venom apparatus was almost certainly there. Wendruff identified what appeared to be a chamber where the animal would have stored venom, making Perioscorpio not just the oldest known scorpion, but potentially the oldest known venomous land-capable animal. From the Silurian into the Devonian and the Carboniferous, scorpions continued to diversify. Some grew enormous. Pulmonoscorpius kirktonensis, a Carboniferous-era species found in Scotland, reached lengths of roughly 70 centimeters, nearly 2.5 feet long, a genuine nightmare crawling through the coal swamp forests of ancient Gondwana. Giant scorpions thrived alongside monster millipedes and dragonfly relatives, with wingspans approaching 75 centimeters, all sustained by an atmosphere richer in oxygen than anything we breathe today. But size was not the winning strategy. As oxygen levels fluctuated and the megaarthropods faded, the smaller, more versatile scorpion lineages persisted. Gradually, they acquired the features we associate with modern species: the preoral cavity for feeding, the refined lungs, and crucially, increasingly specialized venom. By the Triassic period, the crown group of modern scorpions had emerged, with fossil representatives of both the Chactodea and the Buthidia, the superfamily that would eventually give rise to the Indian red scorpion. Every scorpion alive today belongs to one of roughly 22 recognized families. But when it comes to medical significance, one family dominates the conversation so completely that it might as well be the only one. The Buthidae. The Buthidae are the largest scorpion family on Earth, containing approximately 100 genera and over 1,300 species as of recent counts. They are found on every inhabited continent, from the deserts of North Africa to the scrublands of Australia, from the dry forests of Mexico to the tropical lowlands of Southeast Asia. They are, by a wide margin, the most ecologically diverse family of scorpions, occupying humid, temperate, and arid habitats alike. And here is the crucial fact: of the roughly 20 species of scorpion considered medically significant, meaning they possess venom potent enough to kill a human being, every single one, except for a single outlier species called Hemiscorpius lepturus, belongs to the Buthidae. The family has a distinctive set of physical traits. Buthid scorpions tend to have slender, elongated pincers, which are weaker than those found in other scorpion families. Their tails, however, tend to be robust and powerful. This physical trade-off is not coincidental. It reflects a fundamental difference in hunting strategy. Scorpions with large, muscular pincers, like the emperor scorpion of West Africa, rely primarily on brute force to subdue their prey. They crush insects and small vertebrates with those heavy claws and rarely need to sting. Buthid scorpions, by contrast, rely on their venom. Their slender pincers are designed to grab and hold, not to crush. The real weapon is the tail. This is why, as a general rule among scorpion enthusiasts and medical professionals alike, a scorpion with small pincers and a thick tail is far more dangerous than one with big pincers and a thin tail. Among the medically important genera within the Buthidae, the genus Hottentotta holds a prominent and feared position. It is distributed across a vast swath of southern Asia, the Middle East, and parts of Africa. And within Hottentotta, one species stands above the rest in terms of human health impact: Hottentotta tamulus, the Indian red scorpion. This species was first formally described by the Danish entomologist Johann Christian Fabricius in 1798, who named it Scorpio tamulus. The species name was apparently derived from the Tamil people of southeastern India, the region where early specimens were collected. Over the following 2 centuries, the scorpion bounced between several genera. It was referred to as Buthus tamulus, then Mesobuthus tamulus, names that still appear frequently in older scientific literature and even in many modern medical texts. But the correct placement, confirmed by multiple taxonomic revisions, is Hottentotta tamulus. a classification that was actually first established by A. Barriola back in 1914. Though it took decades for the broader scientific community to consistently adopt it in physical terms, the Indian red scorpion is not a large animal. Adults typically measure between 40 and 90 millimeters in length, which translates to roughly 1.5 to 3.5 inches. Its coloration is variable, ranging from a deep reddish-orange to brown and even grayish tones, with dark gray or brownish spots scattered across the carapace and extending down the mesosoma. In 1900, the British zoologist R.I. Pocock distinguished 5 subspecies based on these color variations and their geographic distributions. But modern taxonomy treats these as simple color morphs rather than true subspecies. A red scorpion from Maharashtra and a brownish one from Gujarat are the same species. They simply wear slightly different coats. But do not be fooled by its modest dimensions. This is one of the clearest cases in all of nature where size tells you absolutely nothing about lethality. What the Indian red scorpion lacks in stature, it more than compensates for in the sheer potency and volume of the venom it delivers. Research into the LD50 values, the dose required to kill 50% of test animals, has placed Hottentotta tamulus among the most toxic scorpions ever tested. One study reported LD50 values of 1.3 milligrams per kilogram in juvenile rats and 2.2 milligrams per kilogram in adult rats. Those are extraordinarily low numbers, meaning it takes very little venom to cause lethal effects. And unlike some scorpions that deliver only trace amounts of venom per sting, the Indian red scorpion can inject up to 1.5 milligrams in a single envenomation event. That is roughly 3 times the amount delivered by Androctonus australis, the North African fatal scorpion, which is itself considered one of the most dangerous species in the world. This combination of high toxicity and high volume is a large part of what makes Hottentotta tamulus so lethal compared to other medically significant scorpions. There is also a geographic element to the danger. Unlike some venomous species whose range overlaps primarily with sparsely populated areas, the Indian red scorpion lives in one of the most densely populated regions on Earth. India alone has a population exceeding 1.4 billion people, with hundreds of millions living in the kinds of rural, agricultural communities where scorpion encounters are most common. The sheer number of potential human-scorpion interactions multiplies the statistical risk enormously. The scorpion next door, habitat, range, and living alongside humans, one of the most unsettling aspects of the Indian red scorpion is not what it does, but where it does it. This is not a creature hiding in some remote wilderness far from human contact. Hottentotta tamulus is, by nature, a synanthropic animal, meaning it thrives in close association with human settlements. It does not merely tolerate our presence. it actively benefits from it. The species is distributed across most of India, with particularly high concentrations in western Maharashtra, the Saurashtra region of Gujarat, Kerala, Andhra Pradesh, Tamil Nadu, and Karnataka. It also occurs in eastern Pakistan, the eastern lowlands of Nepal, and more recently, in Sri Lanka, where it was first formally identified in the Jaffna Peninsula in the northern dry zone around 2012 and 2013. The discovery in Sri Lanka was alarming. Prior to that, the peninsula had experienced scorpion stings from relatively harmless species. But beginning around 2006, medical professionals at the teaching hospital in Jaffna began noticing a sharp increase in severe envenomation cases. Local people called the culprit the white scorpion, distinguishing it from the large, mostly harmless black scorpions of the genus Heterometris that were common on the island. After a concerted effort by local and international researchers, the offending species was identified as Hottentotta tamulus, presumably introduced from mainland India, though the exact route of introduction remains unclear. The implications were stark. In 2011 alone, 12 children in the Jaffna district died from confirmed Hottentotta tamulus stings. In 2012, 80 patients were recorded at a single hospital, with 30% of them being children between the ages of 3 and 12. By 2013, stings were being reported at a rate of 4 per week. Why does this scorpion live so dangerously close to people? The answer lies in its preferred habitat and the realities of rural life in the Indian subcontinent. Hottentotta tamulus favors vegetated lowlands with subtropical to tropical humid climates. It makes its home in an astonishing range of microhabitats. A study conducted in the Saswat-Jejuri area near Pune in western India found the species in scrubland with scattered stones, in red and black soil among cropland, on loamy and grassy hill slopes, in mango orchards, inside eucalyptus plantations, and under tree bark with an abundance of over 48%. It was by far the most commonly encountered scorpion species in the study, outnumbering all 5 other species recorded. But it is the scorpion's affinity for human-made structures that creates the real danger. Rural homes in India, particularly in the Konkan region and other agricultural zones, are often constructed from stone, mud brick, or loosely fitted concrete blocks. The walls are riddled with cracks and crevices. Floors are frequently bare earth or roughly paved. Clothing, shoes, and bedding are stored on the floor or draped over surfaces. All of these conditions create perfect hiding places for a nocturnal scorpion seeking shelter from the daylight. Encounters most often occur at night or in the early morning hours, when scorpions accidentally crawl into beds, hide inside shoes, or drop from ceilings onto sleeping residents. This proximity to human families, combined with the scorpion's potent venom, makes it one of the most significant public health hazards in rural India. And the tragedy is that the people most frequently stung, farmers, laborers, women, and children in underserved communities, are often the ones with the least access to timely medical care. Globally, the scale of scorpion envenomation as a public health problem is staggering and largely invisible to the developed world. More than 2.5 billion people worldwide live at risk of scorpion stings. Every year, over 1.2 million people are stung by scorpions globally, and at least 3,250 die as a result. In India alone, scorpion stings are a constant reality in agricultural regions, particularly during the warmer months when scorpion activity peaks and field workers spend long hours in exactly the kinds of environments where encounters are most likely: harvesting crops, overturning stones, clearing brush, sleeping on floors in houses with unsealed walls. The burden falls disproportionately on those least able to bear it. Stings most commonly occur on the extremities, the hands and feet of people working barefoot or reaching into dark spaces without looking first. Many victims first seek treatment from traditional faith healers, locally known as tantrics, who apply poultices, recite mantras, and perform rituals that do nothing to neutralize the venom. By the time these patients arrive at a medical facility, often carried by family members who had to travel miles on foot or by bullock cart, hours have passed. And with Indian red scorpion envenomation, hours matter enormously. Studies have consistently shown that the single most important factor in determining whether a patient lives or dies is the time between the sting and the administration of proper medical treatment. Patients who arrive at a hospital within 2 hours of being stung have dramatically better outcomes than those who arrive after 4 or 6 hours. Every minute of delay is a minute during which the autonomic storm is escalating. The heart is weakening, and the lungs are filling with fluid. Nocturnal hunter, diet, senses, and the mysterious glow. Like all scorpions, Hottentotta tamulus is strictly nocturnal. When the sun sets and the last traces of daylight drain from the sky, it emerges from whatever crack, burrow, or fold of bark it has occupied during the day and begins its nightly patrol. Its primary targets are small invertebrates: cockroaches, beetles, crickets, spiders, and other soft-bodied insects that share its preferred habitats. But the Indian red scorpion is not exclusively an insect hunter. It is also known to take small vertebrate prey, including lizards and, occasionally, small rodents. For a predator that rarely exceeds 3 inches in length, that is an ambitious menu. The scorpion's hunting strategy is primarily ambush-based. It does not actively chase down prey in the way a spider might run across a web, or a centipede might sprint after a fleeing beetle. Instead, it positions itself in a favorable location, pincers extended, tail raised in that iconic forward-curved posture, and waits, sometimes for hours. Patience is not merely a virtue for scorpions. It is a survival mandate. When prey does wander within striking distance, the attack is brutal and efficient. The scorpion lunges forward, seizing the insect or small animal with its pincers. If the prey is small enough and weak enough, the scorpion may simply crush it with its chelae and begin feeding without ever deploying its stinger. Venom is a metabolically expensive resource to produce, and scorpions do not waste it casually. Studies on multiple species have shown that scorpions will often attempt to subdue prey mechanically first, and only resort to stinging when the prey is too large, too strong, or too dangerous to be held by pincers alone. This venom conservation behavior is sometimes called venom metering, and it reveals something important about the animal. Even without a large brain, scorpions make context-dependent decisions about how much venom to inject and when. Once the prey is subdued, the scorpion begins the slow, gruesome process of feeding. Scorpions do not have conventional jaws. They cannot bite and chew in the way a mammal or even an insect does. Instead, they use their chelicerae, a pair of small, toothed, pincer-like appendages near the mouth, to tear the prey apart while simultaneously pouring digestive fluids over it from glands in the midgut. These fluids break down the soft tissues into a slurry of nutrients, which the scorpion then sucks up through a pumping action in its preoral cavity. The victim is gradually reduced to a compact ball of indigestible cuticle, wings, and chitin, which is discarded. The entire feeding process can take many hours. A single large cockroach might keep a scorpion occupied for most of the night. Its sensory arsenal is formidable. Scorpions possess among the most sensitive mechanoreceptors in the animal kingdom. Tiny hair-like structures called trichobothria, arranged along the pincers and legs, can detect vibrations in the air caused by the movement of nearby prey. On the underside of the body, comb-like sensory organs called pectines sweep the ground as the scorpion moves, picking up chemical and textural information about the substrate. These pectines are so sensitive that they can detect the pheromone trails left by potential mates, and males use them extensively during the mating season. Then there are the eyes, and they are stranger than you might expect. Scorpions typically have two median eyes positioned on top of the carapace, and between 2 and 5 pairs of lateral eyes arranged along the front corners. Despite this seemingly generous allotment, scorpions cannot form sharp images. Their vision is blurry by most standards. But what their eyes lack in resolution, they compensate for in raw sensitivity to light. The median eyes of scorpions are among the most light-sensitive in the entire animal kingdom, capable of detecting levels of illumination that would be invisible to most other creatures. They can navigate by starlight alone. And this sensitivity to light connects to one of the most astonishing and still not fully explained phenomena in all of arachnid biology: fluorescence. Every species of scorpion known to science, including the Indian red scorpion, glows a vivid blue-green when exposed to ultraviolet light. Take a UV flashlight into any environment where scorpions live. Switch it on after dark, and the creatures light up against the landscape like small, ghostly lanterns. The fluorescence comes from compounds in the hyaline layer, an extremely thin but remarkably tough coating within the outer cuticle of the exoskeleton. The specific chemicals responsible include beta-carboline and a compound called 4-methyl-7-hydroxycoumarin. These substances absorb UV radiation at wavelengths around 350 to 400 nanometers and re-emit it as visible light in the 450 to 500 nanometer range, producing that characteristic blue-green glow. What makes this property even stranger is its durability. When scorpions are preserved in alcohol, the alcohol itself begins to fluoresce, and scorpion fossils hundreds of millions of years old, with virtually every other part of the cuticle long since eroded away, still retain their hyaline layer and can be induced to glow under UV light. This is a trait that has persisted across almost the entire evolutionary history of the order. But here is the question that has puzzled arachnologists for decades. Why? Why would a nocturnal predator, an animal that actively avoids light and seeks the deepest shadows available, evolve a body that glows under ultraviolet radiation? Several hypotheses have been proposed, and none has been definitively proven. One idea is that the fluorescence functions as a form of UV protection, essentially a biochemical sunscreen. Another suggests it plays a role in communication, helping scorpions locate and identify each other in low light conditions. A third hypothesis, and the one that has gained the most traction in recent years, was proposed by Douglas Gaffin at the University of Oklahoma and further tested by arachnologist Carl Kluk at California State University, Bakersfield. They suggest that scorpions may use their fluorescence to detect UV light levels, essentially treating their entire exoskeleton as a whole body light sensor. When the exoskeleton is glowing, it means UV is present, which means the scorpion is exposed, which means it is time to find shelter. Kluk found experimental support for this when he exposed scorpions to enough UV light to deplete the fluorescing chemicals in their cuticle. Scorpions that could still glow stayed in sheltered areas. Those that could no longer glow wandered more freely into the open. If this hypothesis is correct, then the scorpion's glow is not a beacon or a lure. It is a warning system, a full-body alarm telling the animal that it is too exposed and needs to seek darkness, which for an ambush predator whose survival depends on remaining unseen would be a remarkably elegant adaptation. Newly molted scorpions, incidentally, do not fluoresce. The glow only appears after the fresh exoskeleton has fully hardened, a process that takes hours to days, depending on the species and the body segment. This has led some researchers to propose that the fluorescent compounds are a byproduct of the sclerotization process, the chemical hardening of the cuticle after a molt, rather than something produced independently, whether the fluorescence evolved for a specific purpose or is simply a happy accident of exoskeleton chemistry, remains one of the most captivating unsolved mysteries in scorpion biology. The dance of death, reproduction, and the scorpion's tender side. For an animal so often defined by its capacity for harm, the scorpion's reproductive life is surprisingly gentle and even, in its own alien way, beautiful when mating season arrives, which in tropical regions can span much of the year. Male scorpions begin roaming in search of females. They follow pheromone trails deposited on the ground, their pectines sweeping the substrate with heightened intensity. When a male locates a receptive female, however, he does not simply rush in. That would be suicidal. A female scorpion is a predator, and a hungry one might easily mistake a smaller male for prey. The approach requires caution, signaling, and ritual. What follows is one of the most distinctive courtship behaviors in the animal kingdom: the promenade à deux, a French term meaning a walk for two. The male grasps the female by her pincers, locking his own pedipalp keelly around hers. In some species, he also grips her chelicerae, the mouth parts, with his own in what observers have described as a kind of kiss. And then, pincer to pincer, face to face, the pair begins to move. The male leads, walking backward, pulling the female forward, step by careful step, across the ground. He is searching for a flat, smooth surface on which to deposit his spermatophore, a small, stalked packet of sperm that he extrudes from a gland on the underside of his body. His pectines sweep the ground with increasing agitation as he hunts for the right spot. Meanwhile, the female may resist, pulling away, stinging, or striking with her pincers. In some species, the male responds to this aggression with a sexual sting, a brief, non-lethal injection of venom into the female's body, delivered to the soft tissue of a leg or the mesosoma. Whether this sting functions as a sedative to calm the female, a stimulant to increase her receptivity, or a signal of male quality is still debated. Some researchers have even suggested the sting may deliver a kind of love potion, a cocktail of compounds distinct from defensive venom that chemically primes the female for mating. Once the male finds a suitable spot, he deposits the spermatophore on the ground, then carefully maneuvers the female over it so that her genital opening contacts the sperm packet. The hook on the spermatophore catches the female's gonopore, and the spermatozoa are ejected into her body. The entire courtship and mating process typically lasts between 2 and 15 minutes, though longer durations have been documented in laboratory settings where substrate conditions were imperfect. After mating, the pair separates, and in some species, this is where the male's luck runs out. Sexual cannibalism, in which the female attacks and consumes the male after copulation, does occur in scorpions, though researchers emphasize it is not universal and tends to be associated with species where cannibalism is already a normal part of population regulation. In many cases, the male simply walks away unharmed. What happens next is one of the traits that sets scorpions apart from virtually every other arachnid group. Scorpions do not lay eggs. They are viviparous, giving birth to live young. After fertilization, the embryos develop inside the female's body over a gestation period that varies enormously by species, from several months to nearly 2 years. During this time, the embryos are nourished internally. When the mother finally gives birth, the process can last several hours to several days. The newborn scorpions, called scorplings, emerge pale, soft-bodied, and completely helpless. They immediately climb onto their mother's back, clinging to her exoskeleton in a tightly packed mass. They will remain there for roughly 2 to 3 weeks until they undergo their first molt and their cuticle hardens enough for them to survive on their own. During this period, the mother is fiercely protective. She will defend her brood aggressively against any perceived threat. And despite their tiny size, even newborn scorplings carry venom. Their stingers are functional from birth. After the first molt, the scorplings disperse and begin fending for themselves. They will molt multiple times as they grow, passing through several developmental stages called instars before reaching adulthood. The Indian red scorpion typically lives 3 to 5 years in the wild, though some species of scorpion have been documented surviving much longer under favorable conditions. The venom, anatomy of a chemical weapon, and now we arrive at the heart of the matter. The venom. The venom of Hottentotta tamulus is not a single substance. It is a spectacularly complex cocktail, a mixture of proteins, peptides, enzymes, salts, nucleotides, biogenic amines, and mucoproteins, all working in concert to produce effects that are as devastating as they are precise. A recent proteomic analysis of the Indian red scorpion's venom identified 110 distinct proteins belonging to 13 different venom protein families. The significant pharmacological activity, meaning the components most responsible for hurting you, is predominantly caused by low molecular mass, non-enzymatic sodium and potassium ion channel toxins. Minor toxins make up about 15% of the total venom proteome. To understand why this venom is so dangerous, you need a quick primer on how your nervous system works. Your nerves communicate through electrical impulses, signals generated by the carefully controlled movement of charged particles, primarily sodium and potassium ions, across the membranes of nerve cells. When a nerve fires, sodium channels in the cell membrane open briefly, allowing a rush of sodium ions into the cell. This creates a brief spike of electrical activity, the action potential. Almost immediately, the sodium channels slam shut, a process called fast inactivation, and potassium channels open to restore the cell's resting state. This cycle, fire and reset, is repeated millions of times per second across your body, allowing everything from muscle contraction to heartbeat regulation to sensory perception. The Indian red scorpion's venom attacks this system at its most fundamental level. It contains alpha-type sodium channel toxins that bind to specific receptor sites on voltage-gated sodium channels, known as receptor site 3. When these toxins attach, they physically prevent the sodium channels from closing. The channels stay open far longer than they should, allowing sodium ions to flood into the nerve cells in an uncontrolled torrent. The result is prolonged, repetitive nerve firing, essentially a short circuit in the wiring of the nervous system. Simultaneously, the venom contains toxins that target voltage-gated potassium channels. One of the most well-characterized of these is tamipin, a peptide with a molecular weight of approximately 3,400 daltons. Tamipin is a remarkably specific blocker of small conductance calcium-activated potassium channels, particularly the KCA 2.2 subtype. By blocking these channels, tamipin prevents the nerve cell from returning to its resting state after firing. The sodium channel toxins keep the channels open. The potassium channel toxins prevent the reset. The combined effect is an intense, sustained, and wholly uncontrolled barrage of nerve activity. This dual-action assault on ion channels mediates what clinicians call the autonomic storm. The autonomic nervous system, which controls involuntary functions like heart rate, blood pressure, gland secretion, and smooth muscle contraction, goes haywire. Both the sympathetic division, your fight or flight system, and the parasympathetic division, your rest and digest system, are driven into overdrive simultaneously. The result is a cascade of contradictory signals slamming into every organ system in the body at once. One of the most critical consequences is a massive release of catecholamines, the stress hormones, adrenaline and noradrenaline, from the adrenal glands and sympathetic nerve endings. This catecholamine surge drives blood pressure to dangerous highs, forces the heart into rapid and erratic rhythms, suppresses insulin secretion, causes blood sugar to spike, and constricts blood vessels throughout the body. The heart, suddenly working against enormous resistance with an irregular rhythm, begins to fail. The left ventricle, the chamber responsible for pumping oxygenated blood to the body, can no longer keep up with the demand. Pressure builds behind it, backing up into the lungs. And that is when the most lethal consequence unfolds: pulmonary edema. When the sting strikes, the anatomy of sudden collapse, the clinical progression of severe Hottentotta tamulus envenomation is both rapid and terrifying. It unfolds in stages, and each stage is worse than the last. The first thing the victim notices is pain, severe, localized, burning pain at the site of the sting, usually on a hand or foot. This pain is immediate and intense, and it does not fade. Within minutes, the first systemic symptoms begin. Profuse sweating is almost universal. The victim may begin salivating excessively, drooling as the glands go into overdrive. Nausea and vomiting follow. The skin may become cyanotic, turning a dusky blue as oxygen delivery to the tissues begins to falter. The heart rate accelerates, sometimes dramatically, climbing to 140, 160 beats per minute or higher. Blood pressure spikes. Pupils dilate. Muscles begin to twitch and cramp. In mild cases, this sympathetic storm may peak and gradually subside over the course of several hours, especially if the victim receives medical attention. But in severe cases, particularly in children, the progression is relentless. The cardiovascular system bears the brunt. The venom-induced catecholamine surge causes toxic myocarditis, direct damage to the heart muscle. The heart becomes inflamed and weakened. Echocardiographic studies of envenomated children have shown significant depression of left ventricular function, sometimes within hours of the sting. The weakened heart, already struggling against the vasoconstriction caused by the catecholamine surge, begins to fail as the left ventricle falters. Pressure in the pulmonary vasculature rises. Fluid is forced out of the capillaries in the lungs and into the air spaces, the alveoli, where gas exchange occurs. This is pulmonary edema, and it is the primary killer in Indian red scorpion envenomation. The victim's breathing becomes labored, then desperate. A characteristic pink, frothy sputum begins appearing at the mouth as the lungs fill with protein-rich fluid. Each breath becomes shallower, each gasp less effective. Without aggressive intervention, the victim drowns from the inside. But the venom's attack on the lungs is not purely cardiogenic. Research conducted primarily by Dr. S.B. Deshpande at Banaras Hindu University has demonstrated that there are significant non-cardiogenic mechanisms at play as well. The venom triggers the release of inflammatory mediators, including histamine from mast cells, bradykinin from the kallikrein-kinin system, and prostaglandins. These mediators increase vascular permeability in the lungs, causing further fluid leakage into the alveoli independent of heart failure. A specific high molecular weight toxin, approximately 100 kilodaltons, was isolated from the venom and designated as a pulmonary edema-producing toxin due to its ability to cause lung injury in animal models. The venom also appears to stimulate central serotonin receptor subtypes, contributing to respiratory depression through mechanisms originating in the brainstem. In children, the entire process is accelerated and amplified. A child's smaller body mass means the ratio of venom to body weight is much higher than in an adult. The venom spreads more rapidly through a child's circulatory system. Children are more prone to developing tachycardia, excessive salivation, and encephalopathy. A dangerous swelling and dysfunction of the brain. Clinical studies from southern India have shown that children between 1 and 3 years of age and between 7 and 12 years of age exhibit the most severe adverse effects. In one study of 80 patients admitted to a hospital in Jaffna, Sri Lanka, 30% were children between the ages of 3 and 12. In 2011, 12 children in Jaffna died from confirmed Hottentotta tamulus stings in a single year. The tragedy is compounded by the fact that scorpion antivenom, the immunological preparation designed to neutralize venom toxins, has proven to have limited effectiveness against Hottentotta, tamulus, and venomation. Multiple clinical studies have shown that standard scorpion antivenom demonstrates poor immune recognition and neutralization of the low molecular mass toxins that are primarily responsible for the cardiovascular and pulmonary effects. The antivenom can bind and neutralize some of the larger protein components, but the small, potent neurotoxins that drive the autonomic storm often slip through. This frustrating reality meant that for decades, doctors in rural India were essentially helpless against severe envenomation. They had the antivenom. It just did not work well enough, and that is where the story takes its most remarkable turn. A village doctor's revolution, the Prasasin story. In the early 1980s, a young physician named Dr. Himatro Saluba Bawaskar was working at a primary health center in Burwadi, a remote village in the Raigad district of the Konkan region in Maharashtra. The Konkan is home to one of the densest populations of Hottentotta tamulus in the world, and scorpion stings were a nightly horror. Bawaskar watched villager after villager arrive at his clinic with the unmistakable symptoms of severe envenomation: sweating, tachycardia, cold extremities, and labored breathing. And he watched them die. The mortality rate in the 1970s and early 80s hovered around 40%. In some villages, the death of a scorpion sting victim was considered an inevitable fact of life, as certain as the monsoon rains. Bawaskar had come from humble beginnings, born in a small village to a poor farming family. He had worked every conceivable odd job to fund his education, serving as a waiter, a sweeper, a paperboy, and a bookshop clerk. He faced caste-based discrimination during his medical training in Nagpur and endured periods of depression so severe that he had to take leaves of absence. After earning his medical degree, he chose rural practice not out of convenience, but out of conviction. And when he saw the scale of the scorpion sting crisis in Konkan, he committed himself to finding a solution. He began by compiling case studies and sending his observations to journals. His first individual paper, describing the diagnostic cardiac signs of red scorpion stings, was submitted to an Indian medical journal and rejected. He sent the same paper to The Lancet in London. It was accepted within 8 days and published in 1982. Bowascar initially experimented with sodium nitroprusside, a powerful vasodilator used in hypertensive emergencies, to treat the refractory pulmonary edema caused by severe envenomation. It worked. In 1 month, he treated 65 patients with pulmonary edema, and the results were transformative. But nitroprusside was a dangerous drug. It required careful intravenous monitoring, was unstable in light, and could cause fatal drops in blood pressure if administered too quickly. It was not a practical treatment for a primary health center with limited equipment and a single doctor. So Bowascar went back to the medical literature, searching for something safer. And he found prazosin. Prazosin is an alpha-1 adrenergic receptor blocker. It was originally developed for the treatment of high blood pressure and was also used in pheochromocytoma, a rare adrenal gland tumor that produces excessive catecholamines, the exact same class of hormones that the scorpion venom causes to flood the body. Bowascar recognized the connection immediately. If the pathophysiology of severe scorpion envenomation was fundamentally a catecholamine storm, then a drug designed to counteract catecholamine excess should, in theory, be effective. In 1984, he treated 126 patients with oral prazosin. Every single one survived. He published his findings in The Lancet in 1986, and the paper gained international recognition. The mechanism was elegant in its simplicity. Prazosin blocks the alpha-1 adrenergic receptors that catecholamines bind to in order to produce vasoconstriction and increased blood pressure. By blocking these receptors, prazosin reduces the peripheral vascular resistance that the overwhelmed heart is struggling against. It lowers blood pressure without increasing heart rate. It reduces preload and afterload, taking pressure off the failing left ventricle. It also inhibits phosphodiesterase, leading to the accumulation of cyclic GMP, which further relaxes smooth muscle and improves blood flow. And critically, it can be given orally. No intravenous lines, no pumps, no continuous monitoring. A doctor in a remote village with nothing but a stethoscope and a pill bottle could save a life. Bowascar did not stop at publishing papers. He traveled throughout Western Maharashtra, visiting villages and rural health centers, teaching peripheral doctors how to recognize severe envenomation and how to administer prazosin. He organized seminars, distributed educational materials, and personally trained hundreds of practitioners. All of this work was self-funded. He received no government grants, no pharmaceutical sponsorship, no institutional backing. The results spoke for themselves. Over the following decades, the case fatality rate from Hottentotta tamulus envenomation in India dropped from roughly 40% to less than 1%. That is not a modest improvement. That is a revolution. Thousands of lives, disproportionately the lives of children and agricultural laborers in India's poorest communities were saved because a single physician in a rural clinic refused to accept the status quo. In 2022, the government of India awarded Dr. Himatrao Bawaskar the Padma Shri, the country's fourth highest civilian honor. The Indian Academy of Pediatrics called him an unsung warrior and a living legend. Researchers from Guizhou University in China published a letter in The Lancet praising his work under the title Reducing Scorpion Sting Fatality Rate to 1% in India. It was, by any measure, one of the most impactful pieces of clinical research ever conducted in a resource-poor setting. Venom as medicine, the scorpion's gift to science. It would be natural to view the Indian red scorpion's venom as nothing more than a weapon, a chemical tool of destruction honed by millions of years of predator-prey evolution. But that is only half the story. The same molecular precision that makes scorpion venom so dangerous also makes it extraordinarily valuable to science and potentially to medicine. Scorpion venom has been used in traditional healing systems for centuries. In traditional Chinese medicine, the entire dried body of the scorpion Buthus martensi Karsh has long been used to treat convulsions, spasms, and chronic pain. In parts of India and Africa, folk remedies involving scorpion extracts have persisted for generations. Modern science is now beginning to validate some of these ancient intuitions, not through crude preparations, but through the isolation and characterization of individual peptides with highly specific biological activities. Among the most promising areas of scorpion venom research is oncology. Several peptides isolated from scorpion venoms have demonstrated significant anti-cancer properties in laboratory settings. The mechanisms are diverse and target multiple hallmarks of cancer: induction of cell cycle arrest and apoptosis, inhibition of angiogenesis, the formation of new blood vessels that feed tumors, suppression of tumor invasion and metastasis, and selective blocking of ion channels that are abnormally expressed in cancerous cells. The most advanced application to date involves a peptide called chlorotoxin, isolated from the venom of the deathstalker scorpion Leiurus quinquestriatus. Chlorotoxin has a remarkable property. It binds selectively to the surface of glioma cells, the aggressive brain cancer cells responsible for glioblastoma multiforme, one of the deadliest cancers known. Researchers at the Fred Hutchinson Cancer Research Center and the City of Hope Medical Center have developed chlorotoxin-based technologies for both cancer imaging and treatment. One application, called tumor paint, attaches chlorotoxin to a near-infrared fluorescent molecule. When injected into a patient before brain surgery, the chlorotoxin carries the fluorescent marker directly to the tumor cells, causing them to glow under special lighting. This allows surgeons to see the precise boundaries of the tumor and identify tiny clusters of cancer cells that would otherwise be invisible, dramatically improving the accuracy of surgical removal. Tumor paint has progressed through clinical trials and has been tested in pediatric brain cancer patients with encouraging results. An even more ambitious application uses chlorotoxin to direct chimeric antigen receptor, CAR T cells, engineered immune cells, to selectively attack glioblastoma. Researchers at City of Hope demonstrated that chlorotoxin-directed CAR T cells were highly effective at killing human glioblastoma cells in laboratory assays and animal models without harming non-tumor cells. The first clinical trials using this approach in human patients are underway. From the Indian red scorpion, specifically, the peptide tamipin has attracted considerable research attention beyond its role in envenomation because of its extraordinarily high affinity and selectivity for KCA 2.2 potassium channels. Tamipin has become a valuable pharmacological tool for studying these channels in the brain and other tissues. Research has also shown that recombinant tamipin can induce cell death by apoptosis in human leukemia cells and in certain breast cancer cell lines with remarkably low effective concentrations. While these findings are still in the early preclinical stage, they suggest that the same molecule that contributes to the lethality of the Indian red scorpion's sting could one day be harnessed as a targeted cancer therapy. Other scorpion venom components are being investigated for antimicrobial properties. Several peptides have demonstrated activity against bacteria, fungi, and viruses, raising the possibility that scorpion-derived molecules could become a new class of antibiotics at a time when antibiotic resistance is an escalating global crisis. Still, other peptides are being studied for their potential in treating autoimmune disorders, where their ability to selectively modulate ion channel activity could help fine-tune immune responses that have gone awry. The irony is extraordinary. The same evolutionary process that produced a venom capable of collapsing human cardiovascular systems also produced molecules with the precision to target individual cancer cells without harming healthy tissue. Nature's deadliest chemistry may ultimately prove to be some of its most healing. Conservation, population, and the uncertain road ahead. For all its medical significance, and for all the research devoted to its venom, remarkably little is known about the ecology, population dynamics, and long-term conservation status of Hottentotta tamulus. The species has not been formally evaluated by the International Union for Conservation of Nature. Its population size is unknown. There are no comprehensive surveys tracking its distribution, abundance, or trends over time. This is not because scientists do not care. It is because studying a small, nocturnal, cryptically colored scorpion that lives in cracks and under rocks across a vast and ecologically varied subcontinent is an enormously difficult undertaking. Scorpions leave few traces. They do not vocalize. They are active only at night. And in many of the rural areas where they are most abundant, the infrastructure for systematic biodiversity monitoring simply does not exist. What we do know suggests that the Indian red scorpion is, at minimum, stable in numbers. It is the most abundant scorpion species in multiple studied habitats across Western India. Its synanthropic tendencies mean it benefits from, rather than suffers from, human land use changes. As agriculture expands, as new settlements are built, and as rural infrastructure develops, the scorpion gains new hiding places and new sources of prey. In a grim paradox, habitat modification that harms many species actually favors this one. But this should not inspire complacency. The long-term impacts of pesticide use, climate change, and urbanization on scorpion populations are poorly understood. Broad-spectrum insecticides applied to agricultural fields could reduce prey availability, and fumigation of homes and buildings could directly kill scorpions in their refuges. Climate shifts that alter temperature and humidity patterns could push the species out of some parts of its range, while opening new territory elsewhere. Without baseline population data, we have no way to detect or measure these changes until they become too large to ignore. From a public health perspective, the most critical need is not conservation of the scorpion, but protection of the people who live alongside it. And that means investing in exactly the kinds of measures that Dr. Bawaskar championed: awareness campaigns that teach rural communities to recognize the symptoms of envenomation and seek treatment immediately; training for primary care providers in the proper administration of prazosin, ensuring that prazosin and other essential medications are stocked at rural health centers; building better referral systems so that complicated cases can be transferred quickly to higher level facilities; and conducting research into improved antivenoms that can effectively neutralize the low molecular weight toxins responsible for the most severe cardiovascular effects. There are practical steps that individuals in scorpion-prone areas can take to reduce the risk of stings: sealing cracks and gaps in walls, especially at ground level; keeping bedding elevated off the floor on frames or cots; shaking out shoes, clothing, and towels before use; using mosquito nets not just for insects, but as barriers against scorpions dropping from ceilings or walls during the night; clearing debris, wood piles, and stone heaps away from the immediate perimeter of the home. And perhaps most importantly, knowing the symptoms of envenomation and having a plan for reaching medical care quickly if a sting occurs. The question of whether scorpion sting envenomation should be formally listed as a neglected tropical disease by the World Health Organization has been debated at the highest levels of global public health policy. The WHO's Strategic and Technical Advisory Group for Neglected Tropical Diseases considered the question as recently as 2018. many clinicians and researchers, particularly those working in India, Brazil, and North Africa, where the burden is highest, argue that inclusion on the neglected tropical disease list would bring much-needed attention, funding, and policy focus to a problem that disproportionately affects the world's poorest and most marginalized populations. A survey of Indian medical residents found that nearly 70% supported the designation, citing high prevalence, underreporting of cases, the disproportionate impact on children, and widespread knowledge gaps among healthcare providers. The Indian red scorpion does not ask for our sympathy. It does not need our protection, at least not yet. It has survived for eons without our help, and will likely continue to do so long after the last human argument about public health funding has been settled. But the relationship between this ancient predator and the human communities that share its habitat is one that demands attention, understanding, and, above all, preparation. The Indian red scorpion is not going anywhere. It has survived 400 million years of geological upheaval, climate catastrophe, and mass extinction. It has found a way to thrive in the shadow of one of the most densely populated nations on Earth, and its venom, that extraordinary, terrifying, and increasingly valuable chemical arsenal, will continue to shape human lives in ways both destructive and, perhaps, redemptive. What changes is not the scorpion. What changes is us. Our knowledge, our preparedness, our ability to turn the tools of destruction into tools of healing. The next time you lie awake at night and hear something scratching in the wall, remember this: the creature on the other side of that plaster is older than the mountains, tougher than the deserts, and carrying a chemistry set that modern science is only beginning to decode. It does not want to hurt you. It does not even know you are there. But if your paths cross in the wrong way at the wrong moment, the consequences can be swift, silent, and catastrophic. Respect the scorpion. Understand its venom. And above all, if you live in a region where Hottentotta tamulus is found, shake out your shoes before you put them on in the morning. If this journey into the world of one of nature's most formidable chemical engineers fascinated you, do me a favor and hit that subscribe button. Drop a comment below and tell me what creature you want us to explore next. Share this video with someone who needs to know that a 3-inch scorpion can drop a grown adult in under an hour, and that a single doctor with a blood pressure pill changed everything. Turn on notifications so you never miss a video. And as always, thank you for watching. Sleep well tonight.